Hello everyone and welcome to today's webinar, Correlative Cryo-Imaging for Cell Biology. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems and Aviol. To learn more about our sponsors, please click on the sponsor logo on the left side of your screen. So let's get started. I'd like to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you want at any time you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice you share this webinar on your personal social media. Just click on the social sharing tab and let your friends and colleagues know about today's live event. If you have any trouble seeing or hearing this presentation, simply click on the Support tab found at the top right of your presentation window or report your problem by clicking on the Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Elizabeth Wright. And also joining us for our live question and answer session is Dr. Jay Yang and Dr. Bryan Sibert. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Join me now in welcoming Dr. Wright. Dr. Wright, you may now begin your presentation. Welcome.
Good afternoon. Thank you for joining us for today's webinar on correlative light and electron microscopy for cell biology. We would like to thank our sponsors and co-organizers like Microsystems and Alveo Labs. We would also like to thank LabRoots for hosting this webinar series. As mentioned, my name is Elizabeth Wright. I am a professor at the University of Wisconsin-Madison. Our research group focuses on questions related to infectious disease and human cell dysfunction. We also develop and use a range of cryo-correlative imaging methods for our research. I would like to introduce two other presenters for this webinar and webinar series. Dr. Jay Yang is a cryo-EM biophysicist in the group and develops light microscopy, cryo-EM, and cryo-CLEM methods and software tools. Dr. Bryan Sibert is a structural virologist on the team. He develops and uses light microscopy, cryo-EM, and cryo-CLEM workflows for virology research. To get us started, here's a brief overview of today's webinar. I will introduce cryo-EM at UW-Madison, highlight the major emphasis areas in structural biology and connections to cryo-EM and CLEM. From there, I will discuss some of the theoretical and practical aspects of labeling biological targets and include research examples. We will discuss CLEM equipment options and workflows, including research examples. I will present some of the future areas for CLEM technology development. I will then provide a few suggestions about resources, make some acknowledgements, and point to future events. The Wright Lab moved to UW-Madison in 2018 to begin the next phase of work in cryo-EM structural studies of host pathogen systems. Simultaneously, we set out to build a cryo-EM research center at UW-Madison in the Department of Biochemistry to support the research endeavors of investigators across the UW campus, regionally, nationally, and internationally. We have assembled a team of the PI, 2 staff scientists, Eric and Matt, 4 postdoctoral research associates, Jay, Brian, Julian, and Daniel, and 2 graduate students, Joe and Juan. Members of the group are engaged in facility management and development, research projects, and cryo-EM education. In addition to our team, we have a suite of instrumentation to support all major cryo-EM workflows, from the basics of sample optimization by negative stain, TEM, to single particle data collection and 3D map determination. We specialize in areas associated with cryo-CLEM and cryo-electron tomography, and have built resources to support these studies of biological targets from light-level imaging through cryo-FIT-SIM sample milling, cryo-ET data collection, 3D reconstruction, rendering, and subvolume averaging. The members of the team and the instrumentation are essential for the success of cryo-EM studies in the Wright lab and at the CEMRC at UW-Madison. In structural biology, there are unique disciplines that support investigations of isolated purified complexes through to an entire virus, cell, or tissue. We use techniques that allow us to study the structure and function of the macromolecule or complex at the highest possible resolution and with the highest level of contextual information. Many investigators in these fields are developing the technologies to improve correlation and integration between the methods and the resulting data. Cryo-EM methods, hardware, and computational algorithms have improved significantly over the last few years to support both atomic level resolution structure determination of individual complexes and slightly lower resolution structures of intact entities that provide enhanced contextual information. We are focused on improving technologies that support better correlation between cryo-EM and fluorescence microscopy. So why do we use fluorescence microscopy, correlative light and electron microscopy, and cryo-EM? Briefly, cryo-EM technologies were pioneered to retain specimen hydration and reduce electron beams radiation damage to biological sample while imaged in the vacuum of the electron microscope. These advances have allowed data to be collected that could then be computationally processed to generate native 3D structures of macromolecules. The pioneering efforts and recent advances in the field led to the 2017 Nobel Prize in Chemistry to be awarded to Joachim Frank, Jacques Dubochet, and Richard Henderson. In kind, fluorescence light microscopy was developed for resolving targets of interest during the imaging of live or fixed specimens, in particular biological specimens. Numerous awards, including several Nobel Prizes, have been presented for developments in fluorescence microscopy labeling and imaging technologies. Finally, correlative light and electron microscopy grew from the need to define structures of complexes that were spatially resolved in biological specimens. We can differentiate CLEM methods by whether they produce indirect or direct correlation information. In cryo-CLEM, the goal is to achieve direct correlation data from the specimen. In our laboratory, we develop and use CLEM methods and workflows to support structure-function studies of a range of biological targets. However, while we are able to correlate well between light microscopy and electron microscopy, our goal is to further improve technologies so that there is greater correlation to molecular biology and physiology experiments. We and others are developing tools so that the exact sample that is imaged also produces the essential molecular data. This connection is critical for many reasons. For example, when we image a specimen, we resolve the structure of the virus, for example, and the macromolecules that make it. However, we can only infer from related molecular experiments that the likely number of infectious virus particles on that specimen that we've imaged. If we are able to design pipelines that support one-to-one correlation, we could define the structure and function of a particular virus to ultimately design better therapeutics and vaccines. As we consider the cryo-EM workflow to use, so much depends on the biological question. What is the target? Isolated macromolecules, a macromolecular complex or assembly, whole cells, or whole cells that have been perturbed by a virus or other agent, or whole cells and tissue. At the same time, there is a balance between context and the achievable resolution based on how the sample has been processed and imaged. When we move forward with a project, we consider the workflow in 4 phases: sample optimization and preparation, specimen preservation, where we can use plunge freezing or other high-pressure freezing or self-pressurized rapid freezing approaches, the imaging pathway that we're going to employ, cryo-EM imaging to collect 2D images, cryo-electron tomography imaging to collect tilt series to generate 3D reconstructions, and then conventional EM approaches to understand general ultrastructure of an object. Finally, we consider the computation and data analysis resulting from cryo-EM imaging, cryo-ET imaging, or conventional EM imaging. For this series of webinars, we will focus on the central pipeline as illustrated in this red arrow and these circles for imaging of intact cells for light microscopy, cryofluorescence light microscopy, and cryo-electron tomography. Now, we will highlight some of the labeling strategies that have been used for a range of biological targets. There are several key items to consider when labeling an object of interest. First, what are the options available to the system? Is the system amenable to genetic engineering? If not, is immuno-labeling an option? Or could one develop a new technology for label introduction? I will now highlight these 3 labeling options used for 3 independent biological targets. Increasingly, cryo-EM has become a valuable tool for structure-based drug design. However, due to the more modest resolution gains commonly achieved by cryo-electron tomography, it has not been clear as to if cryo-ET structures could provide feedback for drug discovery and design applications. In this project, we worked with our collaborators to determine if cryo-electron tomography could be correlated with genetic and immunological data to provide validation on an RSV vaccine candidate. For RSV, early work on formalin inactivated vaccines was not effective due to negative impacts on children who received the vaccine. Therefore, for many years, vaccine development was halted until both molecular and structural biology approaches matured. Efforts focused on the development of live attenuated vaccines because one can balance the variables of attenuation with immunogenicity. The other variable that cryo-electron tomography imaging could be used to investigate is the physical nature of the RSV vaccine candidate. Dr. Moore and Dr. Sibert engineered an RSV vaccine candidate using codon de-optimization, as you can see here. The map defines the construct and the alterations that were made. Briefly, on the native virus particles, 3 glycoproteins are present: SH, G, and F. Most human immune responses are to F. The design goal for the candidate was to modify F for increased thermal stability and maintenance of the pre-fusion conformation, eliminate SH, and decodon optimize G to reduce both incorporated and secreted G. We then collected cryo-ET data on a large number of virus particles to assess the structure of the particles and the glycoproteins studying the surface. Here, as we slice through the 3D volume of the tomogram, we note the glycoprotein densities on the surface of the particle, the maintenance of an intact viral membrane, and the inclusion of the ribonuclear protein complex, or RNP, in the interior. At this level of detail, we were confident that cryo-electron tomography combined with immuno-labeling would be valuable for assessing the physical character of this vaccine candidate. For this work and other projects, we developed a native immuno-labeling strategy for surface-exposed complexes because it was necessary for us to have a way to label and identify the structures of interest and also retain the integrity of the virus particles and the macromolecular complexes. Conventional immuno-labeling methods incorporate steps of aldehyde fixation, vigorous washing, and blocking. We knew from conventional light microscopy and EM experiments that a number of these processes damage the ultrastructure and fine structure of virus particles. The native labeling approach was performed under physiological growth conditions and in culture media. The general steps used preserve the virus and cellular ultrastructure. This method could also be used for conventional immuno-EM imaging for greater correlation and validation of results. If fluoronanogold conjugates are used, one can also do CLEM experiments. Our results are shown in the figure panel at the bottom. In A, we see a cell extension with assembling and released virus particles surrounding the periphery. B and C are cryo-EM images of two fields where several RSV particles have immunogold labeling on the F glycoprotein. Panel D is a slice through the 3D tomogram where black arrowheads denote gold particles that are associated with the F glycoprotein. Further quantification of cryo-ET and conventional EM of the immuno-labeled particles allowed us to determine the ratios of pre, intermediate, and post-fusion F present on the vaccine candidate as compared to wild-type virus particles. The results confirmed those of other experiments that there was a greater number of pre-fusion F on the particles, indicating the success of the genetic approach used to produce the candidate. Our laboratory research also centers around structural studies of bacteria and their appendages, namely the flagella and the pili that support cell motility and interactions with abiotic and biotic surfaces. We use Caulobacter crescentus as a model system because of its dimorphic life cycle and the presence of polarly localized flagella and pili that are present during the swimming and swarmer phase of the life cycle. In this study, we and our collaborators sought to determine whether the pili were able to both extend and retract and to quantify the force of retraction for one pilus and multiple pili. Here, in this study, we were also interested in labeling a periodic surface-exposed structure, i.e. the pilus. However, conventional genetically encoded tags could potentially alter the structure of the pilin monomer and its ability to self-assemble into the pilus filament. In-kind immuno-labeling approaches, while effective, could alter the function of the pilus during extension and perhaps retraction. Therefore, our collaborators developed a method for introducing cysteine residues into the pilin monomer. These residues were positioned so that maleimide dyes could associate with the pilins along the length of the pilus filament. In time course, phase contrast, and fluorescence microscopy imaging, pili were observed to extend and retract from the cell poles, as you can see where the blue arrow is present. We followed up on these experiments with cryo-CLEM imaging to study the structure of the cell pole, and the pilus filament during extension and retraction. In this figure panel, we have a cryofluorescence microscopy image in A. Here we see several Caulobacter cells that are autofluorescing in the green channel with the pilus, with the and have the AF594 maleimide dye in the red channel associated with the pilus filament. We collected a series of cryo-CLEM images and transferred those to the cryo-EM for imaging. Panel B shows where we identified the exact same cell in A, and in C, we have a slice through the 3D reconstruction of that cell with a pilus filament extended in that particular slice. The rendered view in D notes the pili in blue and the cell wall structures in orange, yellow, and red. This study was one of the first instances where the Leica-EM cryo-CLEM was used to study bacteria and the structure function of bacterial appendages. Our third example is of how labeling strategies can be effectively used for time course experiments. In this case, our collaborators were interested in determining the timing of and structural changes that occur during the process of HIV attachment, fusion, and entry. They engineered a pseudotype virus that contained a YFP tag on VPR and an M-cherry tag on the GAG polyprotein. In live cell imaging experiments, when the virus particle is intact, the signal from the two labels is co-localized. When the particle fuses with the host cell-associated membranes and the particle contents is released, the co-localized signal is no longer present. Multicolor labeling strategies are very effective. However, in some cases, the events can be sparse, cells may be too thick for EM imaging, and there can be challenges associated with freezing. This is where imaging by fluorescence microscopy and cryofluorescence microscopy can help to resolve these challenges prior to imaging the samples by cryo-EM. The figure panel here walks us through the cryo-CLEM process. Panel A is an overlaid image of the cryo-CLEM map onto the cryo-EM low magnification map. Here we are able to pinpoint regions of interest on the EM grid and in the images and in the collection of navigator files that contain the coordinate information. In panels B and C, we have a series of higher magnification overlays that provide greater information about the grid square, the cell edge, and the labeling of the virus particles on that cell. This region was selected for cryo-electron tomography data collection, and we were able to identify several virus particles in E, F, and G for further analysis. Here I will play a movie of that cryo-electron tomography data set. This movie illustrates not only the power of cryo-CLEM for locating biological targets of interest on large surface areas of the grid and the cells present on a grid, it also shows the value of whole cell cryo-ET for investigating the ultrastructure of cells and the structures of cell components. As we slice through the 3D volume, noted with red arrows are the virus particles, in green boxes are mitochondria, with purple arrows are polyribosomal complexes, and in the purple box is a ribosome-studded vesicle. Also revealed in this data set are long stretches of microtubules and actin networks. Their presence and stable structure are indicative of our success in preparing the samples for the light microscopy and freezing process for downstream cryo-CLEM and cryo-ET imaging. During this next portion of the webinar, I'll provide you with some information about equipment systems and workflow development and designs for cryo-CLEM. We all have really exciting biological questions that would benefit from light and electron microscopy imaging. However, we may not know what resources we need for addressing these questions and whether we should house them in our own laboratories or use them as part of a shared resource. There are a lot of options available for the equipment, and decisions regarding the systems to purchase and used depend on many experimental factors that are generally determined during the initial development of a project, and those are refined over its course. For the ideal and highly automated cryo-CLEM workflow, one will need access to a live cell imaging system, a plunge freezer, cryo-CLEM microscope, a cryo-FIB-SIM for sample pinning, and a cryo-TEM. In the next few slides, I will show some of the results of studies with more modest instrument setups. In this CLEM study, to determine the organization and structure of tether and restricted HIV particles that had budded from cells, we had access to live cell imaging systems, plunge freezers, and a side entry cryo-TEM. This was an effective workflow, and we are able to correlate between the live cell imaging data and the cryo-EM tomography data to resolve the HIV particles and the tethering protein. We were fortunate that we did not lose our points of interest between the time of imaging and plunge freezing or during sample transfer steps pre and post plunge freezing. In a follow-up set of cryo-CLEM experiments, we replicated the previous results and improved correlation to multiple targets by using the Leica cryo-CLEM system post-plunge freezing and prior to cryo-TEM imaging. We were able to rapidly overlay the light microscopy and EM images and transfer the coordinate positions between the cryo-CLEM and the cryo-TEM, which sped up our data collection process. As with the previous experiments, we did have to take care during multiple transfer steps to minimize damage to the specimen via mishandling or sample warming that could cause devitrification. We were limited in our investigations to the periphery of cells due to sample thickness considerations. When one has access to the full set of equipment for cryo-EM and cryo-CLEM studies, limitations associated with time course experiments or sample thickness can be eliminated. However, we still may experience some pitfalls associated with sample damage due to mishandling, devitrification, or other items. Examples of the entire workflow will be presented in a future webinar in the series. So how do we practically do cryo-CLEM? We have developed a cryo-CLEM workflow that includes standard operating procedures in areas that may require optimization to improve the success rate of cryo-CLEM experiments for the novice and experienced practitioner. This workflow is conveniently color-coded to help the investigator work through each step and the associated steps of the process. In order to help individuals plan and troubleshoot whole-cell cryo-CLEM experiments, I will outline a few of the critical steps within the workflow in the subsequent slides. Arguably, one of the most critical items for whole-cell cryo-CLEM success is the substrate onto which the cells adhere. There are many options for the substrates. The most commonly used one is gold grids because they are not toxic to cells. The gold grid is the base upon which an additional support layer is applied, and this is typically, in our instances, a carbon film. The film is necessary for cells to adhere to and spread over the grid surface. When we examine the overall architecture of the grid, we have the options of using a standard mesh, these over here, center marked mesh, or a finder grid. Finder grids are extremely helpful because they have fiduciary markers present that aid in correlation experiments. The style of the carbon film, either solid or perforated, depends on user preference and data collection requirements. When we transition to grid preparation, there are a number of considerations. Briefly, one should have a standard set of tools and laboratory equipment to minimize specimen contamination and continuity of research. We prefer to use MatTech dishes for the experiments because they have a glass cover slip in the bottom that allows for periodic light microscopy imaging without the introduction of grid handling steps that could damage the grid or the cells that are cultured onto them. We may also add an additional layer of carbon to the grid surface to improve substrate stability and increase what we term cell happiness. Finally, we may add, in this stage, an extracellular matrix protein across the grid surface, i.e. on top of the carbon film, to enhance cell adhesion and spreading. Finally, we also add fluorescent and electron-dense markers to the sample to support correlation experiments and image alignment during the reconstruction process. In short, we have had great success with culturing a wide range of cell types on EM substrates for cryo-EM and cryo-CLEM experiments. These include bacterial cells, mammalian cells, both laboratory and strains, as well as primary cells. Cells can be co-cultured together and perturbed with various treatments such as transfection and infection. As mentioned earlier, there are many techniques that may be used to label cells for a correlative imaging pipeline, and we've used a multitude of these. During the initial stages of an experiment, we will image the cells to assess growth and their happiness level, as well as labeling efficiency and cell thickness, as we can see here in this Z stack. If cells do not spread well or are thicker than approximately 750 nanometers, we may consider the use of a sectioning or milling approach prior to cryo-EM imaging to thin the cells sufficiently. Once you've prepared your sample, imaged it by light microscopy, and vitrified it via plunge freezing or high-pressure freezing approach, it is time, yay, for cryofluorescence microscopy imaging. There are many options available for use. The selection of the system depends on the attributes of the particular system as well as the biological target you're interested in. Ideally, one would use a CLEM system that is part of a seamless workflow, has minimal transfer steps between the systems or methods for handling that eliminate possible sample damage. And the imaging and coordinate data is rapidly correlated with another instrument's software. For our work, we have had great success with the Leica-EM cryo-CLEM system for studies of bacteria, human cells, and virus-infected cells. Finally, we consider the TEM that is available at your institution and or through a consortium or a national laboratory. There are two major models, side entry style microscopes and autoloader style microscopes. Both are extremely valuable tools for use in specimen screening and data collection. In many cases, the instruments can be set up for semi-automated or automated data collection routines. We have used both types for our cryo-CLEM studies and cryo-electron tomography studies of cells, and the results from each have enabled us to resolve the biological questions that we were engaged in addressing. So, as with all areas in structural biology, we are looking at the future of CLEM imaging. Are there improvements to the methods and technologies that will lead to better and faster results? Yes, absolutely. Most critically, we should have dedicated patient and creative personnel. For the novice user, we can have well-developed standard protocols for them to follow. We typically focus on, in our lab, the improvement of substrates and cell culturing methods through micropatterning and microfluidics. And we are also engaged in then developing improved automation routines through software and hardware advances. That said, there are many other advances that can be made, including to labeling strategies, additional hardware advances, and there may be other needs to include minimizing transfer steps. In the following slides, I will highlight the areas that we are interested in particular, how to improve cell culture and substrates, and software routine. Now, this is just a preview of a subsequent webinar that will follow. And so as you've noted throughout this webinar, we have a robust workflow for cryo-CLEM studies of virus-infected cells. This workflow relies on tools from Leica, namely the EM-GP and the EM cryo-CLEM system. With these tools in hand, we are developing improved routines for better observation and correlation between cryofluorescence microscopy images and cryo-EM data. In this slide, Jay Yang, a postdoctoral fellow in the lab, demonstrates an improved workflow for correlation between cryofluorescence microscopy and cryo-EM data. The data is of an RSV infected cell where the native immuno-labeled virus particles fluoresce green. You can see here, in particular, in this yellow box. In the cryo-ET reconstruction, we resolved the structure of the virus particle that was identified in the cryofluorescence microscopy map. Of note, the label used was fluoronanogold. We were able to resolve the nanogold particles associated with the F-glycoprotein on the surface of the virus particle. A second really exciting area of development in the lab is micropatterning to support complex cell culturing experiments and improved automation for cryo-CLEM. The leading rationale for micropatterning is that cells in the body do not grow in 2D or in isolation from one another. Therefore, technologies can be developed to support the expansion of native in situ microenvironments on a substrate. This will expand our capacity to understand fundamental cell biology and design experiments to tease out architectural, mechanical, physiological, and other structural and functional relationships present between cells and tissues and organ systems. In the lab, we have a Leica DMI-8 microscope with the Alveol Primo micropatterning system. This is an extremely user-friendly system that allows us to pattern substrates easily for cell culture experiments. The system is also housed in the same room as the cryo-CLEM and plunge freezing instruments. This makes transitions between the three pieces of equipment easy and workflows easily accessible to users. In addition, experimental timelines are on the order of 24 to 48 hours, moving from the micropatterning system and basic light level imaging through to CLEM. Of course, this very much depends on the timing of how long it takes for cells to grow and spread on the EM grids. In a sneak peek for one of the upcoming webinars on micropatterning for cell biology, I wanted to highlight a few different patterns that Bryan generated on the grid surface, transferred to the grid film. He then applied the ECM, that then supported the growth of HeLa cells for subsequent light microscopy and cryo-EM imaging. And here you can see the two different patterns Bryan applied to the grid. One is straight lines covering the grid. They're highlighted in the lighter color. And then the lower pattern is one of a slightly deformed triangle. And in each case, the HeLa cells, as they were cultured onto the grid, uniformly adhered and maintained the coverage over those micro-patterned environments. And finally, when we look at, in G, a cryo-EM image, we can resolve the cell body where it's in the particular spatial arrangement over the micro-patterned region. So we were super excited about this technology and the advances to cell biology that it will support. So many times we ask ourselves, well, what's next? So how do I begin my project? My recommendations are to contact vendors about the latest technologies. This is something I do frequently. Or ask a member of a cryo-EM research facility or a cryo-EM research lab for a consultation and discussions on how to move forward with your project. Many people are available to you in the community, and everybody is super responsive and supportive of one another, and we're happy to answer your questions and assist with your experiments. I've also provided a few reference site materials for LICA as well as Alveol, so you can contact them directly to find out more information about cryo-CLEM technologies and micropatterning tools. At this time, I'd like to acknowledge a few members of the large team that supported these research endeavors both at UW-Madison and Emory University. Initially, I'd like to thank the members of my group, the alumni who have moved on to do other things. They really set their foundation for the lab's development in infectious disease research. The members of the group that developed the initial stages of cryo-CLEM were Sherry, Josh, Rebecca, Dunlong, and Hong. Currently in the lab at UW, Jay, Bryan, and Joe are the ones leading the charge in developing CLEM technologies and micropatterning for our expanded studies in infectious disease and cellular dysfunction. For our biological targets, we had a really great group of collaborators supporting our research efforts in HIV, RSV, and Caulibacter research. Finally, I'd like to thank our vendors from LICA and Alveol. In particular, LICA, we'd like to thank Dr. Louise Bertrand and Ms. Nancy Rizzo, and from Alveol, Dr. Aurélien Dubois and Mr. Serge Kadura. At this point, I'd like to highlight our upcoming webinars in the series. So we have 4 additional webinars. The first webinar will be held on Thursday, July 2nd. And this is a webinar to show how to improve grid preparations for cryo-CLEM, including micropatterning technologies. This is a hands-on workflow webinar where we will share videos of how we prepare the grids and different tips and tricks for improving your capacity to do correlative cryo-EM experiments. Following this webinar, we'll have 3 scientific results webinars that discuss improvements for automation and correlative microscopy workflows, correlative imaging of host pathogen interactions, and advancements for correlative imaging of cell-to-cell interactions. We'd like to thank you for joining us today and your attention, and we'll now pause for questions.
And thank you, Dr. Wright, for that informative presentation. And I want to welcome Dr. Sibert and Dr. Yang. We will now start the live Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So we have quite a few questions already coming in, and I'm going to start with you, Dr. Wright. My tech dishes do not have flat bottom glass compared to Wilco. Can that affect visualization of viruses entering in the cells?
That's an excellent question. In our case, with many of the collaborators we're working with, they've been using MatTech dishes for some time for all levels of fluorescence microscopy work. So we had a lot of confidence in that we wouldn't have any problems associated with being able to visualize the virus entry or events of virus assembly in the samples. So we haven't experienced any difficulties with the MatTech dishes alone or with the samples being on MatTech dishes or EM grids that are then sitting in the MatTech dishes. You can also adjust where your Z-plane is to allow you to focus and follow the events of interest. Thank you for that question.
And thank you. And Dr. Sibert, this next question is for you. Is this micro-patterning technique compatible with all TEM grids?
Yes. So we have used the micro-patterning on carbon-coated gold and nickel grids, as well as silicone grids. We haven't had any grids that it wasn't successful on.
Thank you for that. And Dr. Yang, viruses are very small. Below the wide field resolution, less than 200 how could you be certain that the localization of both fluorophores is specific to the structures observed in cryo condition when fluorescent is not visible in the cryo-EM?
Very good question. So generally, people do several things to actually improve the correlation accuracy as well as the signals they are looking for under the cryo-CLEM. So right now with the Leica system, you can actually achieve theoretically somewhere between like 200 to 600 nanometers resolution. And then that's way below the 200 micron for that question. And then the next part is that you also need to, if you want to achieve really high accuracies for the correlations, you can also use fluor sphere, which is beads. And those beads are generally very small and way much smaller than the point spread functions, the full length and the half maximum densities, that values. So generally, it fell right into the range, and then you can actually image them. If you can get a really good correlation accuracy, you can generally image the particles. A lot of groups have success in the particles with a diameter range from 50 nanometers to 100 nanometers.
Thank you. And Dr. Wright, which step tends to have the most user errors?
Again, an excellent question. Typically, we see the most challenges during specimen handling and transfer steps. This is because we are typically using gold grids, which are extremely fragile, and inexperienced users may inadvertently damage the grid while they're handling the grid with tweezers and other manipulators. So this is one reason why many research groups and vendors are developing workflows that minimize the steps where individual grids, i.e. in isolation, are handled. And they're doing this by preloading the grid into some sort of stabilizing unit, or even more so within a chuck type system that then can be transferred between one microscope to the next. Thank you for the question.
Thank you very much. And actually, Dr. Wright, I'll give you another one. What cell thickness is appropriate for the cells on the grids?
Sure. This is a question we get frequently. So we have had great success with culturing cells of all thicknesses on EM grids. And I should say we are either culturing cells or we're laying down cells on the grids that are separate from one another. So we are not, at this point in time, growing a confluent monolayer. We still want the cells to have separation between one another so that we also have exposed areas of the grid showing, especially if we're doing correlative microscopy, so we can have those points of correlation. In addition, when some of our experiments, we're looking at cell peripheries, so we want to have those regions exposed. So generally, mammalian or human cells are on the order of 2 to 15 microns at their maximum Z dimension. These are cells we're culturing on grids, and this would be around the nucleus. The points to consider here are where you have complete vitrification of the cells on the grid if you're using plunge freezing. And when this is a concern, one may want to use a cryoprotectant in the media prior to freezing, or if the region of interest for your project is really in the thickest portion of the cell, you may want to consider a cryo-sectioning or a cryo-milling technique prior to the cryo-TEM imaging, or if your sample is quite thick, where you would not get complete vitrification using plunge freezing method, you may want to consider high pressure freezing, freeze substitution, and more conventional TEM imaging.
Thank you so much, Dr. Wright. And this next question is for you, Dr. Yang, and it's a two-part question. How many fluorophores can we image with the CRYO-CLEM, and how long does it take to acquire a tile scan of the whole grid?
Great question. So right now, the fluorophores we applied like two types of the fluorophores, depends on our experiments. We apply either 100 nanometers or 500 nanometers. 100 nanometers or like 200 nanometers if we don't want to use the 500 nanometers. 100 nanometers, we use it for the localization, accuracy, estimation, and correlation. And the 5 and 200 nanometers, we also use that to help us do the correlation. And for how long it takes to get a cryo-CLEM, this process, the longest time take is actually for the scanning of the tiles, the entire grid. And then generally it takes 40 minutes, 30 to 40 minutes. It also depends on the Z stacks you choose to collect on each tile. And then we generally, for cells, we use 15 micron. And then also depends on the steps you used. We use a Nyquist, which is 350 nanometers, so you can actually get a better image for later deconvolution processing.
Thank you for that. And Dr. Sibert, what kind of shape geometry can be patterned on the grid?
So, great question. Any shape or pattern that you can think of can really be used during the micro-patterning. The minimum size is going to be determined by the resolution of your particular device. The Primo system has a resolution of less than 2 microns when you're using a 20x objective. And we have routinely done patterns with features as small as 5 microns. One thing you want to think about, though, is you probably want your cells on the carbon support in between the grid bars. So you will want to design a pattern that fits within those single grid squares, which are around 90 microns.
And thank you. And Dr. Yang, I'm going to ask you a question. Before I do, I want to thank our audience because these are just great questions and we have so many coming in. Thank you so much. Dr. Yang, what is the time requirement for learning to be proficient at each stage of this workflow?
Great question. I think it really depends on the comfort levels of the users. Generally, I think right now with the CLEM system, Leica system, it's much easier because the software layout is very clear. I think the majority of the time a user have been spending on would be the sample preparation as well as the loading process. That may take some practice, and then it's generally take probably 5 to 10 rounds to get very comfortable.
And thank you. Dr. Wright, this next question is for you. First, the audience member says, hi, this is Joanne from Clin Nanomedicine, and how to cut the CRYO block into slides for EM without typical EM fixation. And thank you for your presentation, she says.
Sure, Joanne, that's an excellent question. And so it really depends on your particular sample that you're imaging. So in our case, we're doing cells that are cultured in less than a monolayer. And so one of the techniques that has become prominent today, and remember these are cryo-fix specimens, people are using cryo-FibSim, so that's under cryo conditions, so better than minus 150 degrees Celsius. You're inserting that into a scanning electron microscope that has a secondary beam, that's the focused ion beam, and so you can use that to mill down your specimen. And so there's 3 major techniques that can be used that I highlighted on one of the slides. You can either produce a lamella, you can do just sectioning off the top and continue imaging, or you can cut out what's termed a lift out and then transfer that to your grid and then image just that lift out portion. And so there's a recent paper by another group in the field, and we're happy to share references to the literature that has shown using this technique on C. elegans. One can also do cryo-ultramicrotomy to section material and thin it for cryo-EM work. If you needed to do fixation, because that was one of your critical points, you have the prospect of doing fixation or cryopreservation and then subsequent free substitution and embedding if your sample is way too thick to consider doing any cryo-milling or sectioning approach. Thank you again for your question.
Thank you. And this next question is for you, Dr. Sibert. This is, can this technique be used to track GPCR internalization?
I don't have that much experience with the GPCR internalization myself, but if it's something that you can track with fluorescence and electron microscopy, then this technique should be applicable to that as well.
Thank you so much. This next question actually is another question for you, Dr. Sibert, and we have time for a couple more questions. The audience member says, I read an article on cryo-EM structure of the 2019 nCoV spike in the perfusion conformation of RAP et al. 2020. It's interesting that you've studied the HIV chemokine receptors and RSV viruses. How feasible is the application of the CLEM microscopy in the study of the pathogenesis of SARS-CoV-2 virus?
Yeah, that's a great question. And actually, it's very applicable to the study of the SARS-CoV-2 virus, similar to the other studies that he mentioned. I think many groups are already using cryo-EM to study the SARS-CoV-2, and there's already several cryo-EM structures that have published. And this is an exciting use of this technique to learn more about that virus.
Thank you for that. Our next question is for you, Dr. Wang. Can the techniques be used to track GPCR internalization?
The GPCR internalization,
yes.
I'm not very familiarized with the size of that complex, but I think that is probably really small. I guess right now the most success people get is actually from the 50 to 100 nanometers. They can use to track it. I think it's doable under the fluorescent conditioning. It's going to be probably challenging to localize them very precisely just due to the size of the complex.
Thank you so much. This next question is for you, Dr. Sibert. I'm trying to find it. I just had it. Actually, let me just ask this next question, and I'll open it up for whichever one of you would like to answer it. Do you require binoculars on the plunge freezers for freezing adherent cells or suspension of cells on grids using Leica GP or other plunge freezers? And or do other plunge freezers work too?
So this is Liz. I'm happy to take that question. So on our Leica EMGP, we do have the binocular feature incorporated into it. And so this is really useful when we're training new users on how to use the unit so that they can see how their grid is being manipulated and how the filter paper is coming in and blotting it. And so they can really dial in the conditions that we're interested in for manipulating the sample. We don't always use binoculars for visualizing the grids during the process, either on the EMGP or other systems that we have in the lab. It's really, training new people, starting with a new sample, and then once you have everything dialed in, then you can move on to not necessarily checking it every time.
Thank you very much. And Dr. Sibert, this is our last question for today. Regarding the micro-patterning process, are there some limitations with the patterns?
Yeah, thanks. That's another great question. Not really. So again, any shape or pattern that you can make in Illustrator or computer software programs can then be projected onto the grid. And with the Primo system, it can even do different levels of patterning. So you can have areas of your pattern with more ECM adhered than other areas of your pattern. So it's really powerful, flexible technique. And again, I'd say the only limitation is going to be the minimum resolution, which is around 2 microns.
Thank you so much, Dr. Sibert. And I want to thank all three of you, Dr. Wright, Dr. Sibert, Dr. Yang. Thank you very much for your presentation today. Do you have any final comments for our audience before we close?
Hi, Sarah. Thank you for asking that. Well, I think our final comment would be just reach out to your vendors, your local EM experts, and others in the community, and ask questions like you've asked us today. We're sorry we couldn't address all of them, but we will happily address them offline. And we will, it's just, you have to try. There's no limit to the technologies and being able to use EM, cryo-EM, and light microscopy to resolve a variety of different biological questions. Oh, and stay tuned for our next webinar in the series, which is really a virtual hands-on webinar where we will show you some of the steps of how we actually manipulate cells on the grids and use micro-patterning and walk you through the steps of the process so that hopefully we're providing you with some confidence that you can go out and test drive these experimental techniques on your system of interest. We really appreciate your time today.
And we appreciate your time. Thank you again, Dr. Wright, Dr. Sibert, Dr. Yang, for your today and for your contributions to cell biology. And I'd also like to thank LabRoots and our sponsors Leica, Microsystems, and Aviol for underwriting today's educational webcast. Before we go, I want to remind the audience, thank you for joining us today and for your interesting questions. And questions we didn't have time for today and those submitted during the on-demand period will be addressed by our speaker via contact information we provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. Until next time, take care, be safe, bye-bye.